Hi everyone, my name is Lindley and today I'll be discussing phylogenetics through a phylogenetic tree based on an inanimate object. Before we dive in, I want to explain phylogenetics. Phylogenetics study the evolutionary relationships of organisms by way of comparing populations or species. Such relationships get illustrated through phylogenetic trees, which clearly show the genetic similarities or differences of these organisms. It is important to note that not all trees look the same, as many of them have different looks depending on what is getting illustrated. You can see an example of this here. The groups that get included in these phylogenetic trees are referred to as operational taxonomic units and can be anywhere between whole species or individual organisms. Hopefully this video will help you better understand this concept through an example using writing tools. Before we dive into my example, I want to begin by explaining a couple of things that hopefully will make this easier to understand. Here is the phylogenetic tree we will be using, illustrating different writing tools. Each writing tool which will reside on one of the tree branches and will represent an operation taxonomic unit. This phylogenetic tree is considered to be rooted since all the writing tools are derived from a common ancestor, identified here as the gray square. The triangle points shown here are known as the internal nodes and are important because they are indicative of the most recent and common ancestor of each tree clade. Now for my example. As you can see, all the writing tools are paper oriented except for the dry erase marker found at the top. This dry erase marker must have initially originated from a common ancestor, therefore making it the outgroup within the tree. The first divergence here evenly separated the colored dry erase writing tool from all the other black writing tools. The next divergence separated markers from pens. The Sharpie and Crayola are both markers which tells us that a recent common ancestor obtained this trait, making them closely related to one another. The next node splits the writing tools between having a thicker fiber tip and having a thinner tip. The felt tip pen is a cross between the traditional pen and a marker, creating a larger dark line, while all the other pens have a thin tip. The types of pens that evolve from this split are the fine line and thicker line pen types. The fountain and fine tip pen both obtain a very small tip that create for incredibly thin lines, helping us establish that they must be genetically similar to one another despite their differences. Whereas the gel pen, the ballpoint pen, and the rollerball pen have a small tip but produce a thicker, less fine line, making them also genetically similar. As indicated with the blue arrow here, the longer lines within the phylogenetic tree indicate an early divergence, whereas the red arrow pointing to the shorter lines indicates a more recent divergence. Before we conclude, two terms that are important to know are cladogenesis and anagenesis. Cladogenesis occurs when one lineage gets split into two and the branches evolve without one another. Anagenesis occurs when changes happen within one lineage and happens in every species. An example of anagenesis can be seen with a single brand of the felt tip pen, as over time this type of writing tool has changed and evolved, whether it be from ink type, tip size, materials, etc. Its lineage has changed. Without either of these processes, a lot of what we know and have today in many aspects of life would not be what they are. For the final time this semester, I hope you found this video to be helpful and informative. Thanks for tuning in.